Welcome to this targeted oncology presentation in precision medicine called TRK inhibitors, a tumor-agnostic targeted therapy. Hi, I'm Dr. Shubham Pant, a medical oncologist in MD Anderson Cancer Center in Houston, Texas. I'm here with my colleague, Dr. David Hong, Deputy Chair of the Department of Investigational Cancer Therapeutics and Associate Vice President of Clinical Research, also at MD Anderson. Targeted therapies have really come to the forefront of the treatment for cancer. Today, we are going to talk about an important new therapeutic class that has shown great promise amongst several advanced malignancies called TRK inhibitors. These agents represent a paradigm-changing advance in precision oncology and drug development for rare mutation-driven cancers. Dr. Hong, welcome to the program.
Well, thank you for inviting me, Shubham.
That's great. So first tell me, this is kind of a brand new kind of science in oncology. This is kind of a very different tumor-agnostic approach which has come to the forefront nowadays in oncology. These new drugs have been recently approved. So first tell me, what are these tropomyosin receptor kinases? What are these TRK? What is it?
It's a really good question. These receptors have been known for a long time, and in the normal human functionality, they serve as really receptors to modulate neurologic functions. For example, TRK is involved in things like temperature regulation, pain. TRK has a number of other functions. It helps regulate movement. To some extent, it regulates weight. Wow. Lastly, TRK is oftentimes involved in proprioception. And it wasn't until the 1980s when they first identified that certain cell lines, colorectal cell lines, actually had TRK fusions that would really constitutively and oncogenically make these cells drive because of these TRK fusions.
So what you're saying is that in this gene, so the genes, what are the genes which actually have these fusions, and how does it lead to protein creation?
The genes are TRK1, 2, and 3.
Okay.
And what ends up happening is that in the development of the cancer cell, there's an actual fusion or a translocation where the 3' end of the NTRK gene then fuses in a way with the 5' portion of another a partner gene, and therefore leads to a receptor that is in a way constantly on and drives and is a growth signal for that cancer cell.
So what you're saying is that you have the fusion and that kind of turns a switch on, which is normally off, and that kind of causes these cells to divide. And then they create the proteins also, right? The TRK A, B, and C proteins?
Correct. And this signals through a number of oncogenic pathways, such as the MAP kinase pathway and also PI3 kinase, et cetera.
So normally, so this is when they're dysregulated in humans and everything, I guess, that it's used for normal human development, but then when they're dysregulated, then they can lead to cancer. So that's very interesting. So tell me, how would you inhibit these pathways? What is the best way to inhibit these pathways if it is, if in humans and everything it's causing cancer, how would you inhibit these pathways?
So what others have discovered is the best way to inhibit these pathways is to really inhibit that ATPase pocket, these aberrant TRK receptors used to, drive the signaling. And so recently, obviously we'll be talking about larotrectinib and entrectinib, but there have been drugs, other previous drugs that have sought to target that ATPase kinase pocket.
And that's on the cancer cell. You're saying the fusion is on, so the switch is on, it's creating this protein, so you're trying to block it at the level of the tumor cell, yes, right? Okay. So people were made these inhibitors to target that exactly pocket, and what does it do? If it fits into it, what does it lead to?
Well, it leads to inhibition of that ATPase pocket. Therefore, there's no downstream signaling of that signal or of the receptor.